Yes, thank you very much for the invitation to Chicago, and I will tell you about our recent research on co-translational processing of Gp5 and Gp3 of Arteriviruses. And first, I want to give a very brief introduction what happens during synthesis of glycoproteins. So all starts with a signal peptide, which is present at the N-terminus of glycoproteins, and this targets the ribosome with a nascent protein chain from the cytosol to the ER. And here the signal peptide is integrated into the translocon, which is a polysubunit transmembrane protein through which these pores, the proteins, are then translocated into the lumen of the ER. And here two enzymes are waiting. First is the glycosyl transferase, which attaches carbohydrates to N-glycosylation motifs, and this is called the sequon. And the second enzyme is the signal peptidase, which cleaves or does not cleave the signal peptide from the protein. And usually uncleaved signal peptides anchor proteins to membrane. So it's a quite different outcome for a protein whether a signal peptide is cleaved or not. And little is known about the interplay between signal peptide cleavage and glycosylation, because both of them can act on a nascent protein chain. And especially for arteriviruses, there are glycosylation sites and signal peptide cleavage sites located in close vicinity. So the first part of the talk will be about Gp5 protein. As you all know, it forms a complex with the M protein, and it is mainly responsible for budding of virus particles. And Gp5 and M, as you have just heard, is a target of neutralizing and non-neutralizing antibodies. And you also have seen this classical slide of the course of the immune response in infected pigs. So virus causes viremia after 2 or 3 weeks, and the virus persists for at least 5 months in lymphatic tissues. And when you look for the antibody response, the total antibody response appears very early after infection, but these antibodies made in the beginning are not neutralizing. Neutralizing antibodies appear later, and they may be helpful to clear the virus from the blood and finally from the body. So the question is, why do neutralizing antibodies appear only late during the course of infection? And several years ago, a model was put forward which states that a decoy epitope in Gp5 might explain persistence of PRRS. So Lopez and Osorio identified 2 epitopes in Gp5. One is a neutralizing epitope and the other is a non-neutralizing epitope, but this epitope is immunodominant, meaning it can act as a decoy. Thus, when antibodies are bound to this decoy epitope, the other epitope might be shielded by neutralizing or for neutralizing antibodies. But when you look at the primary structure of the protein, you can see that the decoy epitope might be located in the predicted signal peptide, so just dependent on where and if the signal peptide is cleaved, the decoy epitope might not be present in the final protein. So we first asked, is the signal peptide cleaved in Gp5 from PRRS of the American VR2332 strain, and is cleavage dependent on N-glycosylation, because between those epitopes there's a hypervariable region with changing number of glycosylation sites. So Bastian Tarr in the lab, he made several mutations. So he exchanged glycosylation sites at position 30, at position 33, and he also inserted an additional glycosylation site at position 34, and then he expressed the complete Gp5 gene equipped with an HA tag in CHO cells, and in Western blot you can see, based on the size of the protein, that the glycosylation site N30 is not used. So when you remove the site, the protein has the same size as a Gp5 wild-type protein. In contrast, when you remove the site N33, the protein becomes smaller, meaning this glycosylation site is used. And when he inserted an additional glycosylation site, the proteins has a lower STS page mobility, meaning this glycosylation site is used. And then, to look for signal peptide cleavage, one has to deglycosylate the Gp5 molecules, and you also need a marker, a size marker, with a defined composition. So Bastian synthesized the same construct, Gp5HA, in vitro in reticulocyte lysates, and there you get the complete protein, including the signal peptide, but without glycosylation. And then when you compare the size of this in vitro synthesized protein with all the other wild-type and mutant Gp5, you can see that all proteins run faster than our marker protein, meaning the signal peptide is cleaved in each case. So it's independent of glycosylation in the vicinity. And we also tested Gp5 from various other PRRS strains and also analyzed signal peptide cleavage in various cell types and always got the same result. So now we know that the signal peptide is cleaved, but we do not know at which site it is cleaved. Therefore, we used mass spectrometry to identify the precise cleavage site. And for that, you need a lot of protein. So the Gp5 was cut out from gel in which virus particles were separated, and also Gp5 was recombinantly expressed in insect cells, and these bands were cut, excised from the gels, they were digested with chymotrypsin, and peptides were identified by mass spectrometry. And first of all, no peptides which correspond to the N-terminus, to the signal peptide of Gp5 were identified, meaning that the signal peptide is cleaved in each case, so there's no fraction of Gp5 which retains the signal peptide. But Eberhard Krause, with whom we collaborate on that, he identified two cleavage sites. So one of part of the fraction starts here, and this is then called site 1, and Gp5, which is cleaved here, retains the decoy epitope, and another fraction of molecules is cleaved at site 2, which is just behind the decoy epitope, meaning that another fraction of Gp5 molecules does not contain the decoy epitope. But the disadvantage with mass spectrometry is that it's not quantitative, so we cannot tell whether the main fraction contains or does not contain the decoy epitope. Therefore, Bastian again made new mutations. So the idea was to allow cleavage of Gp5 only at one site, and the signal peptide usually requires small amino acids at position -1 and -3, and to get a completely uncleaved Gp5 molecule, the alanine was converted to a large phenylalanine at site 1, and also alanine at site 2 was converted into a large phenylalanine. To get a protein which is cleaved only at site 1, site 2 was mutated in a similar manner, and to get a protein which is cleaved only at site 2, the site 1 was equipped with large and hydrophobic amino acids. And here again you see the SDS page mobility of all these mutants after deglycosylation. So this is a wild type protein. This is this mutant which is indeed, which does contain an uncleaved signal peptide. This is the SDS page mobility of the protein which is cleaved only at site 2, and this exactly corresponds in size to the wild type protein, and the protein which is cleaved at site 1 is only partially cleaved, but you can see a small difference in the SDS page mobility between the protein which is cleaved at site 1 and the protein which is cleaved at site 2. And therefore we think that the major fraction of Gp5 molecules is cleaved at site 2. And to summarize this, so a major fraction of Gp5 is cleaved at site 2, and therefore the decoy epitope is removed, and probably a minor fraction of Gp5 is cleaved at site 1, and the decoy epitope is retained in this case. And interestingly, there's also microheterogeneity at known conserved glycosylation sites. So N30 is not used in every fraction of the molecule, and also N33 is not used in every molecule. So we have a large variation in Gp5 molecules present in virus particles. So the second part of the talk deals with the Gp3 protein, and the following experiments were done with the equine arteritis virus, but in the end I will give some reason that the results also apply for PRRS. So Gp3 is present in a complex with Gp2 and Gp4, and both of those are typical type 1 membrane proteins, and they form a disulfide linkage already in the endoplasmic reticulum. In contrast, Gp3 is a very unique protein. It forms a disulfide linkage with one of the other proteins only in budded virus particles. And the membrane topology of Gp3 is not completely resolved, so there are two models. In one model, the N-terminus, which is believed to be an uncleaved signal peptide, anchors the protein to the membrane, or in model 2, both the N-terminus and the hydrophobic C-terminus anchor Gp3 to the membrane. This shows a primary structure of Gp3, so it's an N-terminal hydrophobic signal peptide, a hydrophobic region at the C-terminus, and 6 used glycosylation sites. And it has been reported that the signal peptide of Gp3 is not cleaved. However, when you do this signal P, which is usually a very reliable bioinformatic tool, it predicts cleavage with high probability. But these bioinformatic predictions do not take into account protein modifications. They are just based on the amino acid sequence. And when you look at the amino acids in the vicinity of the signal peptide, you can see an overlapping sequon and NTT. And therefore, our working hypothesis was that glycosylation of this overlapping sequon adjacent to the signal peptide inhibits signal peptide cleavage. So Anja, she made mutations of one, the other, or both glycosylation sites. And here in this Western plot, you can see that both sites are used. So here both asparaginins are exchanged and the protein becomes smaller. Here only the first and the second glycosylation site is exchanged, and again the protein becomes smaller. So the overlapping sequon near the signal peptide is glycosylated at both sides. And to look whether this affects signal peptide cleavage, the proteins were again deglycosylated. And here again you have a marker for a protein containing the signal peptide but without glycosylation. And this has the same size as Gp3 wild type, meaning that the signal peptide is not cleaved, as already has been published. But when both glycosylation sites were removed simultaneously, you can see the protein runs faster, meaning that after removal of glycosylation sites, Gp3, the signal peptide becomes glycosylated. So removal of 2, but not 1 glycosylation sites allows signal peptide cleavage, and this is independent of the cell type on the tag which we have attached to Gp3 and on the mutation. So we had different exchanges of asparagine to glutamine and also to histidine, but both allowed signal peptide cleavage. So this is a summary of the first part of Gp3. So glycosylation protects a potentially cleavable signal peptide. So glycosylation occurs first during translocation, and therefore signal peptidase has no access to the site. But when you exchange the glycosylation sites, signal peptidase can cleave the signal peptide. So this seems to be somewhat at odds with the previous models for the topology of Gp3, because N-glycosylation usually cannot occur in the vicinity of the membrane. So when we assume that the uncleaved signal peptide acts as a membrane anchor, it's hard to understand how carbohydrates can be added very close to the membrane. So usually from other studies, it's known 8 to 10 amino acid space must be here until a carbohydrate can be added. Therefore, we decided to analyze the topology of Gp3. So we fused the yellow fluorescent protein to the C-terminus, and if it's exposed to the cytosol, then the YFP would be accessible to proteases in transfected cells. This shows fluorescent pictures of Gp2 YFP, Gp4 YFP, where also the C-terminus was equipped with YFP. And after opening the cell membrane and adding of a protease, you can see that the fluorescence is gone, meaning YFP is exposed to the cytosol, which confirms that Gp2 and Gp4 are type 1 membrane proteins. In contrast, Gp3, here the YFP is protected against protease attack, and therefore the YFP and also the C-terminus of the protein is exposed to the lumen of the ER. Does the signal peptide mediate membrane anchoring, and what happens if the signal peptide is cleaved by removal of the 2 glycosylation site? This shows the result of a membrane binding assay. So we prepared microsomes and separated the microsomes in the membrane fraction and in the luminal fraction, and we had 2 controls. First, influenza hemagglutinin. This is a membrane protein, and this is present only in the membrane fraction. We had a control for a luminal protein, which is called reticulin, which is present mainly in the luminal fraction. And when you look at Gp3 YFP, this is a wild type, this is a mutant with a cleaved signal peptide. You can see both are present in the membrane fraction, meaning that cleaving of the signal peptide from Gp3 does not get a soluble luminal protein. So Gp3 with and without signal peptide are both membrane bound. Cleavage of signal peptide does not affect membrane association of Gp3. So next we deleted the hydrophobic C-terminus from Gp3 and looked whether the protein becomes secreted into the supernatant of transfected cells. So this is a complete full-length protein, wild-type protein with cleaved signal peptide. There's nothing secreted in the supernatant. In contrast, when the C-terminal hydrophobic domain was deleted, you can see that both protein with cleaved signal peptide and the wild-type protein are secreted into the supernatant. So this means that the C-terminus anchors Gp3 to the membrane. So what happens if you introduce the same mutation into equine arteritis virus particles? One would assume that if you convert a protein with a cleaved signal with an uncleaved signal peptide into a cleaved one, this would be detrimental for virus growth, but this was not observed. So both the wild-type protein and the mutant with the two deleted glycosylation sites grow to similar titers in BHK cells. And in the Western blot from virus particles, you can see that after removal of two glycosylation sites, Gp3 runs faster, meaning also in the context of a virus infection, the signal peptide is cleaved from Gp3 after removal of two glycosylation sites. And we did many other studies, so the mutations are stable upon passage. The protein composition of virus particles is identical, meaning that signal peptide cleavage from Gp3 has little, if any, effect on virus replication in cell culture. In contrast, we could not make a mutant where the C-terminus was deleted, so we can conclude that the C-terminus, in contrast to the signal peptide, is essential for virus replication. And this is then the new model for the membrane topology of Gp3. So I think that the signal peptide is not a membrane anchor, but slips out of the translocon during translocation, and therefore carbohydrates can add it here to the site, and this prevents signal peptide cleavage, and the hydrophobic C-terminus anchors the protein to the membrane, but it's not a transmembrane region because the C-terminus is exposed to the lumen of the ER. And one can speculate what could be the function of protecting a signal peptide by glycosylation. So one can speculate during the course of an infection, the signal peptide might be cleaved, for example, because glycosylation sites are lost, or in certain cell types, maybe the signal peptide is cleaved because signal peptidase is faster than glycosyl transferase, and this would create a protein with different antigenic properties, and this might be, for example, also contribute to persistence. What about Gp3 of the other arteriviruses? So in equine arteritis viruses, 66% of all Gp3 molecules have two of these glycosylation sites. 33% have just one site, and there are only three sequences that have no glycosylation site. And also in all PRRS strains, there is one glycosylation site adjacent to the signal peptide, and also in LDV, there is one glycosylation site adjacent to the signal peptide. And when you do the signal IP prediction tool, you can see that all signal peptides are predicted to be cleaved, and for one protein, for Gp3 of LDV, it has already been shown that the signal peptide is retained in the major protein, and therefore I believe that this unique processing scheme of Gp3 might be conserved in all arteriviruses.